This is the second prep. Just going through now the real unraveling of the genetic code. So where we finished, we had figured out what the first four codons were. These were the easy ones because they just took poly U and found out that UUU encodes phenylalanine, and they did the same with A, G, and C to get those codons um, where all the nucleotides in the triplet are the same. Okay, so that was the easy part. But then the next step is to mix two nucleotides together. And you can mix them in equal amounts, and that will give you some information, but you can also mix them in different amounts. And so the idea is you do the same experiment as the experiments with just a single nucleotide, but you take two different nucleotides. And these will be put together randomly by the enzyme. Remember, the polynucleotide phosphorylase is not going to use a template or anything, so it just grabs whatever nucleotides are near it and puts them together. And so all possible codons for those two nucleotides will be made, and the proportions will vary depending on whether there's more G than C. And how much more. You'll be able to, you can do calculations to predict how much of each codon you should see. There's no preference for any particular combination, so it just depends on the amount of nucleotides of each type. And then you can do experiments to find out which amino acids are present and what the approximate percentage is, and make a connection between the proportion of a certain codon and the same proportion of a certain amino acid. So that's the idea. And I'm just going to go through a very simple example, and then I'll show you the results of a more complicated one. And the kind of calculations I would ask you to do would be more like the more simple one. Okay, so this is it. So suppose we took C and A nucleotides, ribonucleotides, of course, not DNA nucleotides, and we put them together in a 2-to-1 ratio, and we have a huge amount of them, like a vast excess, so that incorporating the nucleotides really is truly random. And so what it means is that in the tube, 2/3 of the nucleotides are going to be C's, and 1/3 of them, the rest of them, are A's. And so when we're trying to figure out the chance of any particular combination, is going to be the product of the probabilities. So the idea is, what is the chance that the enzyme grabs a C? It's going to be 2/3. And then what's the chance that the next nucleotide it adds is a C? That's going to be 2/3. And then what is the chance the next one is going to be a C? And that's also 2/3. And then the next one is an A, maybe that, that's 1/3, and so on. And so as the molecule is being put together, at each point, there's a 2/3 chance of adding a C and a 1/3 chance of adding an A. And so you can work out the probabilities this way. So if you have all C's, it's going to be 2/3 for the first C, times 2/3 for the next one, times 2/3 for the next one. And if you remember how to multiply fractions, 2 by 2 by 2 is 8, 3 by 3 by 3 is 27. And then what if there are 2 A's and 1 C? Well, the chance of the A is 1/3, and the chance of the next A is 1/3, so we multiply those. And then the chance of the C is 2/3, and so that is 2 in 27, right? And so if you are ever asked to calculate the probability of every single different possible codon, if you have two different nucleotides, you have 8 possible codons total. So you should know that. So you should know that there should be 8 in total. And then when you do finish your calculation and you add them up, they should add up to 1. If you've done it correctly. And this is just a note. You're going to be solving problems in this class, more so towards the later sections, but even in here you can see that you'll be solving problems and doing calculations. And one of the best ways of being sure you're on the right track is making sure that, in this case, the probabilities add up to 0. If you calculate the likelihood of all of them, then the total has to be 1, right? 1 is 100%. And so it's always good to just check your work. And you can often, suppose you just punch the wrong thing into your calculator one time, you can just quickly skim through your results and make sure they add up, and then that can alert you something is wrong, right? So let's just go through the full calculations for this. So the way I approach it is I take them in turns. So the first possibility, first of all, I know that there's going to be 8 codons. So you have 2 in combinations of 3, which is 2 cubed, and that is 8 codons possible. But if you're not sure, you can just write them all out, right? So they could all be C's, right? So C, 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 that's one possible combination, and we've already done that. And it turns out that is a codon for proline. But you could have 2 C's and 1 A, and there are 3 different combinations for that: CCA, CAC, and ACC. So those are all those combinations. They have the same probability each. And then you could have 1 C and 2 A's, so CAA, ACA, and AAC. So there's 3 of those. And then you could have all A's. And I find that if I'm asked to do, or if I'm asking myself, assigning this to myself to do, this is the way that works out for me when, for example, I'm coming up with an answer key for a question that I've made, right? And then I always check it. So 8/27ths for all C's, and then there's 3 of these. So there's 12 and 27. There's 3 of these. That's 6 and 27, and then there's just 1 of those, 1 and 27. When I add those together, 27/27ths, that's 1, so that's good. Now, if you were doing this experiment, then what you would see is that a total of 12/27ths, or something like around 40%. My mouse is not working very well, would be proline. So we already know that this codon is for proline, and so we're expecting this proportion to be proline. So when we do the actual experiment, suppose we're just trying to figure out how this works, and we see that there's this much more proline, we know that one of these, we don't know which one, because we don't have any other evidence, but we know that one of these encodes proline as well, right? So we have some information. And then if we looked at histidine, we would find that histidine, there's this proportion. So we would know that this is one of the 2 C's and 1 A. And then threonine, this codon and this codon, so we would see that it was 6/27, so we would know it was one of these and one of these. Hopefully you see where we're going with this. And then having 2 A's and 1 C, we'd see 2/27ths of glutamine and 2/27ths of asparagine. And again, we don't know which is which, but we know what that it's 2 A's and 1 C. And then we already knew what all 3 A's was. And so we would get this information, and we could, and what we could infer from that is, okay, proline has 3 C's, or it has one of these, 2 C's and 1 A. And then threonine has, one of these and one of these, and so on. And then you start manipulating the amounts more and see which ones go up and which ones go down. Suppose you made it 5/1 C to A, right? And so on. And then so you would have to do a lot of different experiments. And you would use this kind of information to work backwards and figure out from the amino acid proportions that you saw, and the proportions of codons that must be in the tube, and try to make a connection between those. So you can see that it was not trivial. It was a huge amount of work, because the work of figuring out the amino acids was time consuming and difficult, and then having to repeat the experiment different times with different proportions so you could see which ones increased and which decreased. And then another type of experiment that I'm going to show you a couple of slides beyond this, that was really key to sorting out some of them. Okay, and so this is an example of mixing the experiments, sorry, the nucleotides differently. In this case, it's the A and C as well, just like the previous example, but in this case, there's 5 times more C than A. Okay. And so this just shows you the possible combinations and how much there is of each. Now this presents them in percentages because when you look at the amino acids in the experiments, it tells you percentage-wise what's in the tube. But I would like you, when you're doing the calculations, it's just easier to grade if you leave them as these fractions, okay? Yeah, and so hopefully, like you should go through this. It'll be in your notes, and you can sort of see where these calculations come from, and then sort of see how you can infer again from this the different, what kind of base composition leads to the different amino acids. Okay. Now, we're going to talk about Gobind Khorana. He was a Nobel Prize winning scientist. And interestingly, there's a local angle here because he used to be at UBC. And apparently he was a little grumpy and intense, and they didn't give him tenure, if you can imagine. So he went off to a different university and proceeded to win the Nobel Prize for the importance of his work. And what he did was, instead of combining the nucleotides singly like this, he said, okay, we can only go so far with this. There's still ones where, we know what the composition probably is, but we don't know exactly what the which ones are the correct codons. And so we can sort this out by making dinucleotides. So what you do is you make lots of molecules, for example, that are GU 5 prime 3 prime, and then you use that same enzyme to just glue them all together. And what they do is they form these long strings of GU, GU, GU. And then basically, if you do that same in vitro translation process, and half the codons will be GUG, and the other half will be UGU, right? And that will be valine and cysteine. And if you combine this information with information from experiments like this, that rather than, A and C were actually using U and G. So you had, 5G and 1U, for example. And then you tried to figure out the various codons and stuff, and you could use this kind of a table, except of course with the correct amino acids for G and U combinations. But then if you knew, for example, that a codon was most likely 1G and 2U's, and then from here you realized that you had valine and cysteine here, and you already had decided that cysteine should have 1G and 2U's, then you can pin it down that this is going to be the right codon. And if you've done the same for valine, you've just decided it's 2 G's and a U, but you don't know what the order is, and you get valine out of this experiment, then that tells you that that's the order. So what this helped to do was to pinpoint the exact codons based on knowing the exact order of the codons in this experiment. And so, this is a very important scientist who did really important work. So eventually everything was figured out, and these are the main key points. It's quite a simple code. It's read in triplets. The triplets are referred to as codons. So AUG, for example, is the codon for methionine, and CCC is a codon for proline, and so on. So that's important terminology. The reading frame is established by a specific codon, which is a start codon. That's in vivo, which means in the organism, not in a test tube. You can get it to start without a start codon in a test tube, but in the body it requires the start codon. And the end of the sequence is signaled by stop codon as well. And the code is universal. So there are a few minor deviations, but basically how the RNA is read to produce protein is the same in all organisms. As I said, there's a couple of unusual usages by a couple of minor issues, deviations, I guess we'd call them. But the cool thing is you can take a human insulin gene and you can clone it into a bacterium. And if you have the correct, signals, gene signals in there, it will make human insulin and it will be translated properly. And so this is really a cool feature of this process. And the code is also degenerate. Degenerate doesn't mean it has poor morals. In fact, what it means, what it refers to, is the fact that in many cases there are multiple codons that specify the same amino acid, and the largest is when there are 4. So leucine and arginine both have 6, did I say 4? I meant 6 different codons. So there's 4 here and 2 here. 4 here and 2 here. And then there's a lot, the serine also has 6, right? There's a number that have 4 different codons. And there's a lot that have 2 different codons. And there's very few that have only 1. So trip is one of them, and then methionine is the other one. I think those are the only 2 that just have 1 codon. So that's what degenerate refers to. It means that you can incorporate a serine into a protein with any of these or of these 2 codons, right? But it's also unambiguous. So that means that CUU always encodes leucine. It doesn't sometimes encode proline or glutamine or something. Unambiguous means it always gives you the same amino acid, and it would be really hard to do any kind of experiments if that wasn't the case. The codons that specify amino acids are called sense codons. Remember, I talked about the sense strand when I was talking about transcription, and sense refers to the messenger RNA sequence, which gives the information for the amino acid. So this is just a term to know. But there are 3 different ones that encode stop codons. So these are ones that when this is encountered by the ribosome, that's the signal to complete translation and don't go any further. You don't want to be adding extra amino acids to your protein. You want it to stop in the right place to produce the correct protein. And then one of them is important for the start of translation. And so the start codon is AUG, which is methionine. In bacteria, there's a special methionine called formal methionine, or FMET. And it's a special one for the beginning of the protein. Yes, and so just it's really important to keep in mind that the start and the stop codons are in the DNA and the RNA sequence. They are specific for translation. So the start and the stop codons don't have anything to do with the beginning of either DNA replication or transcription. It's only what is read by the ribosomes to make the protein sequence. And that's it. We will see you in class.